All right, let's go ahead and let's continue this presentation. We left off on a little bit of a cliffhanger because we ended up talking about how do they actually make antibodies against these surface proteins that they have identified by the mass spec differences between the labeled cells with the different hydrogen and deuterium, the heavy and light from the previous figure. Well, this is what the figure that they have in the 2017 paper. They talk about how they generate the antibodies this way, and this is where the students, you guys should see something that looks familiar. You have the phage display on the left side, part B of the figure. And so you can look at this, but this should look familiar to how they do things. Now on the left side, make sure that you see that they have just an antigen, the ECD, which is the domain that will recognize the domain that is actually randomized. And they do a little bit of complicated stuff with a proteolysis site, and then they have the FC domain, then they have a biotinylation tag. This is so it works all with their ELISA. And I'd like you to look a little bit at this just because you can see the biotinylation. But the main thing is the purple Pac-Man thing on the top. You see on the right, that's where it's binding the actual, that's the antigen, I should say. And what they're randomizing is they're randomizing the FAB, the variable fragments, with just the 4 domains with the variable loops on the end, the nice little unit that is one arm of the antibody, and that is what they are actually randomizing. Now, there's a little more detail. I don't get into it. The one thing that they mentioned in the paper, I don't want you to miss this, they say they use an old antibody scaffold that expresses very well. That's one advantage that James Wells brought to this project. He had tried a bunch of FAB scaffolds in the back, and he found that one worked really well with phage display. And so he's using that same scaffold, and then they're randomizing the loops. That's what makes this all possible. It's little things like that that are between the lines of a paper like this. So notice how they had the FC domain. They're using that. This is not the antibody side of things. The antibody side is on the phage. But this is on the beads that they use, which has the antigen on it. So this is a lot of biotech going on. And you gotta, you want to look at it and try to get straight a little bit of what's going on. But this is where the Antibot page comes in. This is where I want you to take that handout, the one that looks like this, and I want you to put this, look at this, and you can see, if you open it up, you can see this nice phage display figure right here. And this is actually in less technical language, so you can actually see a little more about what's actually going on. And this is similar to Delano and Wells. You can't see this from the original paper. You have to dig down to find this. But this is where they actually talk about how they use a reduced genetic code, and they're able to randomize some, but not all residues. That's one of the main things here. And the theoretical diversity is what they can screen, like how many different mutations could coexist using these different codons. And so I think this is pretty cool. This is some biotech nerd detail. But the main thing to notice is that there's only the loop residues are being mutated here. Everything else is staying the same. And so the other thing is, how do they measure binding? And they don't want to do something fancy like surface plasmon resonance. You can do that if you're doing things one by one. But to do things high throughput, you want something simpler. And so what they do is something that is just based on 96 well plates and pipetting. And that's what ELISA is. And if you look at this part of the paper, it mentions ELISA by name. Phage ELISA followed by DNA sequencing. They take what binds according to ELISA, and then they sequence its DNA. And that is one round of selection. Then they will do other rounds of selection, against the one against each target, and they figure out, what sequences of antibodies will bind to these 7 antigens. Notice that they had to narrow it down quite a bit to even do this. They did 7 unique targets that they looked at, and at the end of the day, 2, maybe just 1, were the ones that actually gave good antibodies that they found against it. So check this out. ELISA is the key to how they measure which of their randomized antibodies on the phages will bind. And ELISA is widely used because it's pretty simple. It stands for enzyme-linked immunosorbent assay. And what it means is that you adsorb an antigen onto the surface of a well, the bottom of a plate. You have to block the other sites. That's a technical detail. Then you incubate the yellow antibody, which is the antibody against the thing that you are doing. Now, in traditional ELISA, slightly different from what Wells did. Traditional ELISA, you aren't doing phage display. Instead, you have to detect which well has your binding activity in it. And what they do is they have a secondary antibody. That's the one in green. And the secondary antibody binds the Fc region of the primary antibody. This way, you can actually use a lot of different primary antibodies, and you also do not have to make a special conjugate of the enzyme to the Fc region of your special primary antibody. Instead, you can have a sort of generic secondary antibody that binds all the primary antibodies from a certain species. So you have your anti-rabbit secondary antibody. You're using rabbit antibodies as yellow. And so you throw in your anti-rabbit secondary antibody. That's the one with the enzyme on it. Because it's hard to link an enzyme to an antibody, you don't want to waste your special binding antibody if you don't have to. And so that's it's done for expense reasons and practicality reasons to have two antibodies in this case. Once you have the two antibodies bound, that means that you have the enzyme is attached through a number of other linkages to the bottom of the plate. You wash the plate off, the enzyme's still attached, right? You throw in substrate to all the wells, and the ones that turn the color, the substrate is converted in color by the enzyme. So the ones that turn color are the ones that have enzyme. They're the ones that have the secondary antibody, they're the ones that have the primary antibody, and therefore they are the ones that have the antigen. It's a lot of steps, but it works really well because notice all you need to do is detect color. You need to have a plate, and you can do this in small little things. So this is an example. For example, this shows up all over the place. This is similar to this kind of test that you see in a CNE News publication here. And you see that here's a very simple representation of how you can have antibody binding to a surface. And this is an antibody test that uses a secondary fluorescent antibody, just like the ELISA does. This is like ELISA. It's not exactly ELISA, but I believe it's a sort of streamlined version of it. And so then you shine a light on it and you see if you have the color. And where you have the color, you have the antigen. This is what a typical ELISA assay looks like. Wherever you see the color, you can tell that you have the antigen in that well. And you can also use it on a gel. So if you have an SCS gel that looks like this, that's nice. You have a band of the right molecular weight, but are you really 100% sure that it's the correct protein? If you want to be 100% sure, you add a specific antibody that has and you stain it so that it's the only thing that shows up, and you see that antibody is binding at the exact same molecular weight. And in fact, it's binding better the more pure you get the fraction. That is a good positive result, that not just it's the right molecular weight, but it's definitely the right protein out of the complex gel lysate that you have purified. And of course, then these are things that people do all the time. Probably some of you are taking cell biology and have done this, maybe done it in the lab, and this is it results in bad jokes from people who make Western gels trying to get you to buy their gel, because you can see results with reagents from Thermo Fisher Scientific, one beautiful band. You know, you don't get frowny bands, or a bad antibody will actually bind to multiple bands and it'll give you multiple bands of binding. A good antibody binds to one band and one band only, and you have that nice and pure out of your entire mixture. So, yeah, now you can get another kind of scientific joke once you know what ELISA is. ELISA is related to immunoblotting. And ELISA can be done on a big scale. There's a large-scale proteomic screening where you have a bunch of different experimental conditions, and then here's sera from an adult that has never been infected by malaria on the left, and then an adult who was living in West Africa and has been exposed to malaria multiple times, has developed immunity against it. And they basically put their antibodies, they put their antibodies over a plate that had 4,500 proteins from the malaria parasite. And you can see there are all these proteins that are recognized on the right by the immune system of the person from West Africa. the immune system can learn a lot, and it contains a history of where you grew up and what diseases you've been exposed to. I just want to point out that if you are talking about CRISPR in another class, we don't have a CRISPR journal club this year in this class. We just ran out of days. But there is a CRISPR part to this because they weren't happy to just do their mass spec, their Silas or whatever it was, technique. They also looked for functional surface proteins using a CRISPR screen, and you can look at more detail about that if you want to. There's a long story about that, but we actually aren't going to go into that. I want to focus on the part that's about phage display. And both the CRISPR screen and their mass spec screen both showed the same two hits. They came up with CDCP1 and ITGB5. And the thing is, out of both of those, it looks like they kind of drop ITGB5. They don't talk that much about it, but CDCP1 actually has been found in other cancers. And it is the one where they want to decide it was the strongest lead to go after, to really validate it, to try to go after it with other techniques, and to make sure that the protein you found with this one technique is actually working in the clinic or in an in vivo experiment. And that's where the figure 5 things that we had showed up. That's why they were working with pancreatic cancer, I believe, because it's part of that. So now we're full circle. We finished the paper. The one other thing I want to bring out is there's all sorts of things coming up. This is one of their collaborators. Wells is not on this paper, but Sidhu is a collaborator, a frequent collaborator of Wells, and uses the antibot. And I believe that you see the antibot showing up behind the scenes in a lot of phage display type of things. Here's one where they took this sequence right here. When you see that this sequence has a particular sequence, and in the middle you see a lot of glycines and serines. And what that is that's a linker between two domains that are just for the single chain IG domain. And I just want to point out that this is the entire sequence that they optimized using phage display against a particular antigen. And they're proposing that this is like a really streamlined backbone, like a template that you can use to make a protein that binds another protein. This is what it looks like, and it's actually they made a chain that actually will go from one domain to another so that it will make this one thing as a simple little unit. You see how it's just the 4 domains rather than the 12 domains of the antibody. And one of the things they did is, I always like a nice size exclusion graph because I do a lot of it. And here's where they did size exclusion of their construct. So they designed this new construct. They wanted to show it was stable in solution. And they ran it through size exclusion. And you can see there's a few other bumps, but you get a main bump exactly where you want to get it. You get, it looks like pretty good results for especially considering that they made this big long linker and they cut up the protein and they made this small little streamlined thing. So here they're trying to make the case that this is a possible streamlined form of an antibody that is a single chain of an antibody, and it's all linked together like that. I should say that, yes, this is kind of some heavy-duty protein chemistry that you're talking about here. Once you start to mess around with antibodies, you can design the binding sites, so you can also make linkers to link different domains and make different combinations of the different domains and different parts of the antibody. And then you can do things like, what about if you did a bivalent human VH domain? This is an example where they actually took this and they made an antibody that looks like this. Now this should look weird to you. They just took a single chain, a single domain with 3 CDRs on it, and they put those 2 CDRs onto a functional 4-domain FC. This looks really weird. It only has 6 IG domains rather than 12. It's just got hands, basically, on hands on top of the regular FC domain. But if this binds, it's a more streamlined version of the antibody. And so that's how they put it all together. And again, for this construct, they ran this through a size exclusion column. And you can see, yeah, it's coming out. And you can see from where the standards come out that this is coming out right about at half the size of a regular antibody. Regular antibody will be about 150 kilodaltons. This is coming out at about 75. And that's exactly where you'd expect it to get out because it's half the size of a regular antibody. Maybe this will be a good reagent. It'll be easier to make in the lab. Who knows? The other thing they did is they did binding curves. So here's Biacore binding curves. And the one thing I want you to notice here is they have a equilibrium constant of binding. And it's tight binding. It's nanomolar binding. So they see good binding of this to the receptor binding domain. So it looks like their little streamlined antibody here is actually working. To really prove it, what they did is they did electron microscope analysis, and you can see the spike domain is sort of shown. They show the backbone of that, and they just show little colored lumps where the red colored lumps are where you see add the antibody and you see the extra electron density. So you see it's binding 3 times to the spike trimer. That makes perfect sense. They compare it to where the ACE2 domain binds, where the where the interaction that causes infection, where that binds, and it looks like it overlaps. And so it looks like this could be a reagent where you could add the antibody, it binds like the red stuff over there, and it blocks the blue stuff from binding, and it could be a useful drug. They actually modeled where the blue stuff and the red stuff overlap, and this is what you want to see. The antibody could actually block the entry to it. And then they took it and they used it on hamsters. And it worked in hamsters. They show you the tissues. If you're a biologist and you want to look at tissues, there they are. They looked at whether it gets to the serum or the lungs, how much of it gets to, and it looks like their red dots have nice high concentrations in these different places. And I believe that this is a lung delivered. Let me see. I don't see for sure, but I believe that this might be delivered through the lungs rather than through injection. Wouldn't that be cool if we could deliver a monoclonal antibody by breathing it in? This is definitely a step in that direction, and they're showing that it's actually causing the virus to be, the infection to be blocked and the lung damage to be stopped. So, but of course, as all of us are well aware, SARS-CoV-2 mutates. So once you have this antibody that binds the receptor domain, if it works well enough, it puts evolutionary pressure on the domain to mutate, or for a mutated version of the domain to survive, technically. And so you have a case like this where we had variants appear. And the big question, every time a new variant appears, does it still bind the same antibodies? If you have a vaccination that taught your immune system how to recognize a spike that was from the alpha, does that still work on Omicron? You know all the questions, and there's a lot of dynamics about it. Mutations can cause increased transmissibility, or they can cause immune evasion. And this can measure immune evasion if you look at how antibodies bind, where they bind, and you can even possibly predict if this is going to be a bad thing or not. Not perfectly, but you can at least say it looks like it's a bad mutation, or it looks like it's going to be okay, depending on the type of amino acid change you have and the location that it's at. So that's what this is about. This is one of the first studies in March 2021 about how multiple variants will actually interact. And these are the first couple variants. This, of course, doesn't have Omicron because it was just kicking around in South Africa, or that's where it was first detected. It probably evolved somewhere else. But you can see that you have the wild type, you have good cross-neutralization. As you get more mutants, you get poorer and poorer cross-neutralization until you get things where you have the antibodies don't bind anymore. The more spike mutations you have, the more chance you have that the antibodies are not going to bind. And this makes sense. You see all those spike mutations, you see where antibodies bind. One of them is going to hit where the antibody binds, and the spike will still work, but the antibody won't. And of course, this is what we are all worried about. But when people were all worried about it, I was always a little less worried than other people. I was still worried about it, believe me. But I was also like, well, this is only half the story. Antibodies are only half the story of immunity. There's another half of the immune system. And that is what parts 4, 5, and 6 are going to be about. And so next week, thanks for sticking with it this far, next week we will get around to talking about T cells, which is nature's way of looking inside the cell And as you can expect from our story right here, T cells can also be activated against intracellular tumor mutagenicity cancer changes. So, that will be for next week, however. See you then.